Welcome to this explainer. Today, we're unlocking the fascinating world of biotechnology principles and processes. You know, if you've ever found yourself wondering how on earth scientists literally rewrite the code of life to create life-saving medicines and just totally revolutionary materials, well, you are in exactly the right place. We're not doing a standard dry science lesson today. Nope. We're taking a step-by-step look into the microscopic factory where modern miracles are actually built.
Okay, let's dive into this. Here's our roadmap for today. We're going to define modern biotechnology, meet its early pioneers, open up the molecular toolkit, walk through the exact steps of recombinant DNA, and finally, look at how we scale the whole thing up for mass production. Let's get to it. So, part 1. What exactly is modern biotechnology? We're going from baking bread to genetic engineering.
When you think of the word biotechnology, it's super easy to picture, a hyper-futuristic lab. But honestly, humanity has been using microbes for centuries to do simple things like bake bread or ferment wine. Today, though, we're talking about something way bigger, large-scale, deliberate genetic modification. To really set our boundaries here, let's look at how the European Federation of Biotechnology officially defines it. They call it the integration of natural science and organisms, cells, parts thereof, and molecular analogs for products and services. I know that's a mouthful, right? But in simple terms, it's basically the ultimate merger of biology and engineering. We're literally hijacking nature's own capabilities to manufacture exactly what we need.
To pull off this kind of marvel, we really rely on two twin pillars. First up is genetic engineering. This is the actual physical alteration of genetic material, like DNA and RNA, to fundamentally change a host organism. It's the core code writing, if you will. But then there's the second pillar, bioprocess engineering. Look, the real microscopic world is messy, right? It's full of weeds and contaminants. Bioprocess engineering is all about maintaining a completely sterile, absolutely perfect environment so that only the microscopic workers you actually hired grow, and they grow in massive quantities. You absolutely need both of these pillars to make modern biotechnology work. Moving on to part 2, pioneers of genetic engineering. Let's meet the architects of recombinant DNA. You know, every great architectural feat needs a visionary architect, and our microscopic factory is definitely no different. Let's put some faces to this science. It really starts with Herbert Boyer. He was born in 1936 and wrapped up his graduate work by 1963. Fast forward to 1969, and he was doing this incredibly vital research on restriction enzymes within E. coli bacteria. And this brilliant work caught the eye of a Stanford scientist named Stanley Cohen. Cohen was busy working with these tiny rings of DNA called plasmids. The real spark, the game-changing moment, happened in 1972. Boyer and Cohen actually teamed up, combining their unique discoveries from entirely different fields to successfully construct the world's very first recombinant DNA. Just incredible. And this brilliantly illustrates why Boyer's initial discovery was such a massive catalyst for an entire industry. He observed that restriction enzymes could cut DNA strands in a really particular fashion. It left behind what became known as sticky ends. Think of these sticky ends kind of like biological Velcro. Because they naturally want to form hydrogen bonds with their exact matching counterparts, it gave scientists the unbelievable power to physically cut and paste life's code for the very first time. I mean, wow.
All right, let's jump into part 3, the molecular toolkit. These are our factory assembly parts.
So to build anything on a microscopic scale, you're going to need highly specific, incredibly reliable tools. In fact, there are exactly 5 key tools required for recombinant DNA technology. We've got restriction enzymes, polymerase enzymes, ligases, vectors, and the host organisms themselves. And make no mistake here, this is a strict recipe. If even one of these 5 tools is missing from our toolkit, the entire assembly line completely shuts down. Let's zoom in on those restriction enzymes for a second, our molecular scissors.
Restriction enzymes actually belong to a larger class of enzymes called nucleases, and there are 2 distinct types you need to know. First, exonucleases. These guys remove nucleotides from the very ends of the DNA strand. But endonucleases, they are absolutely the stars of the show. They act as precision scissors, literally snipping the sugar phosphate backbones at highly specific palindromic positions within the DNA. And it's exactly what it sounds like. Just like the word race car reads the same forwards and backwards, a biological palindrome tells the enzymes exactly where to cleanly make that cut.
Now, what's really interesting about this slide is the PBR322 artificial cloning vector you see right here. Notice that ORI section? That stands for origin of replication. Basically, any piece of alien DNA linked to this sequence can replicate and multiply within the host cell. You'll also spot the markers AMP-R and TET-R for antibiotic resistance.
And this is just a brilliant trick. Scientists literally douse the cells in antibiotics, and because of those markers, only our successfully engineered cells survive and keep growing. It's kind of ingenious when you think about it.
Moving right along to part 4, steps in recombinant DNA. Let's hit the assembly line.
So the crucial point here is that it all comes down to a very precise 6-step factory floor process. First up, we have the isolation of DNA, basically breaking open the cell. Second, fragmenting that DNA using those restriction endonucleases we just talked about. Third, isolating the exact DNA fragment we actually want. Fourth, ligating or pasting that fragment into a vector. Fifth, transferring it all into a host organism. And finally, step 6, culturing those host cells on a massive scale so we can extract the final product. Let's take a closer look at how on earth we pull off step number 3.
To isolate the exact fragment we want to clone, we use this visually striking technique called agarose gel electrophoresis. See, DNA fragments are negatively charged molecules, right? So we force them toward a positive anode through a natural seaweed-derived gel. And it's basically a race. The smaller the fragment, the farther it travels. Once they're separated by size, we stain them, and honestly, this is where the magic happens. Under UV light, this invisible microscopic world suddenly glows with these brilliant, bright orange bands. It allows scientists to literally pluck out the exact genes they need. How cool is that?
Step 5 is transferring that DNA, and the delivery mechanisms here are pure sci-fi. DNA is hydrophilic, meaning it loves water, so it won't just easily slip past a cell membrane. To force bacteria to take it up, we have to make them competent. We do this by treating them with calcium and giving them a sudden heat shock. For animal cells, we might use direct microinjection right into the nucleus. And for plants, get this. Scientists literally shoot the cells with DNA-coated gold or tungsten bullets using a biolistic gene gun. I mean, a gene gun. We can even hijack disarmed viruses to act as sort of biological delivery vehicles.
Now, sometimes, before we can even begin all of this, we need a much larger sample of our desired DNA. Enter the polymerase chain reaction, or PCR for short. It essentially acts like a biological photocopier in 3 steps: denaturation, primer annealing, and extension of primers. And here's a super surprising fact for you. The special heat-resistant DNA polymerase enzyme used in this process, it was actually stolen from a bacterium called Thermus aquaticus, which happily lives in boiling hot springs. That is exactly why it survives the lab's extreme high-temperature denaturation step without breaking a sweat.
The results of this PCR cycle are just staggering. By repeating this process over and over again, we can take a single gene of interest and amplify it roughly 1 billion times. That's a billion with a B. This gives researchers an incredible abundance of the genetic material they need to proceed with the whole assembly line. Okay, let's move to part 5 and see how this builds, scaling up and processing for mass production. So, we've successfully engineered our cells at this point, but to make a real-world impact, we absolutely have to transition from tiny microscopic test tubes to massive industrial output. Imagine a gigantic, high-tech brewery, right? But instead of brewing beer, you're brewing millions of dollars worth of life-saving vaccines or insulin. That's exactly what these simple and sparge-stirred tank bioreactors do. Processing anywhere from 100 to over 1,000 liters, these massive vessels provide a continuously stirred, perfectly oxygenated, and strictly sterile environment to cultivate our engineered cells on a true industrial scale.
But the manufacturing process absolutely doesn't end inside the bioreactor. To get a finished usable product, it has to undergo downstream processing. This is the ultimate safety net, and it's absolutely crucial. It involves the careful separation and purification of the product. Honestly, this is the entire reason the medicine you pick up at the pharmacy actually heals you without making you severely sick from leftover bacterial byproducts. After all that, it undergoes incredibly strict quality control testing and clinical trials to ensure it's completely safe and effective for the market.
We've just traced the entire biological assembly line, literally from the microscopic action of a single restriction enzyme all the way to these massive industrial bioreactors pumping out life-saving medicines. It's truly a marvel of modern science. So, I want to leave you with this thought to ponder today. How will these profound, cut-and-paste molecular techniques continue to shape the future of human health, agriculture, and the biological world at large? The potential isn't just breathtaking, it's actively rewriting the future. Thank you so much for joining me on this explainer, and keep exploring.